Uh, yes, good evening, everyone. I hope I'm visible and audible to everyone over here. Just give me a thumbs up in the chat box if I'm audible and visible to everyone over here. Yeah. Ma'am. Just let me know in the chat box. Yes, so that I can start with the class, right?
Sir, please share the screen.
Okay, done. I think the audio and visuals are perfectly fine.
Yes, sir. Yes, sir.
Okay, perfect. Sure. So yes, welcome to the session. Yeah. Okay, welcome to today's session on the carbohydrate part 2, where I will be discussing the glycolysis part, right? The citric acid cycle and gluconeogenesis. I will also try to cover the isomerism of carbohydrates, like in the form of very short answer questions, right? So now, without wasting much more time, guys, you already know the glycolysis, the TCA cycle, and the gluconeogenesis are very potent topic for from the exam point of view. Like they're being asked like 10 marks questions, right, directly. So how to write that particular answer in your answer sheet, I will be discussing that also. But before that, let us guys, just I want to tell you about this that yeah, this is about the Oracle app, right? So the in this basically session, the exam mastery live classes, I will be revising the important topics along with you in a very no concise conceptual manner, right? So all the volatile important topics I will be quickly revising with you guys, right? So that is very much beneficial for your university exams, right? So you can use my code Dr. Abhijeet 20, right, for the discounts, right? So yes, so let's start with the carbohydrate metabolism part 1, right? Now, overview of carbohydrate metabolism. Now, guys, just understand this. I'm telling you, if you understand the overview, the other things becomes very easy. So in spite of ratifying the cycles, just try to analyze the concept first, right? So guys, you already know that yes, in the stomach, we are consuming carbo. Okay, in our body, we are taking the carbohydrate rich diet, right? We are taking carbohydrate rich diet. Now you guys already know the carbohydrate that we are consuming, it is in the form of disaccharide. It is in the form of disaccharide, right? So obviously in the stomach, this carbohydrate or the disaccharide will go and it will undergo a process that is simply called as the digestion, right? It will undergo digestion, right? It will undergo digestion. Now, you already know after digestion, what are the products that will be formed. Now, guys, you already know that yesterday we have discussed that there are three major disaccharides that we are consuming. That is the maltose, we have sucrose, and we have lactose. So we have three disaccharides that we are majorly consuming in the diet. Now, this maltose will be broken down into glucose plus glucose, right? Now, glucose is a monosaccharide. Glucose again is a monosaccharide. The sucrose, again with the help of enzyme sucrase, will be broken down into glucose plus fructose. Glucose plus fructose. And lactose will be broken down into glucose plus galactose. Glucose plus galactose, right? So okay, maltose with the help of enzyme maltase will be broken down into simpler carbohydrate units. Sucrose with the help of enzyme sucrase, lactose with the help of enzyme lactase. Now, a very simple question that I wanted to discuss with you guys that carbohydrates gets digested to form the carbohydrates gets digested to form not only glucose, guys. You can see along with the glucose, fructose is also formed and galactose is also formed. Majority majority of the times you guys are writing only glucose. So carbohydrates get digested or they're broken down to glucose, also fructose, and also galactose, right? Also galactose. So we have three components, right? Now after digestion, okay, we have three components over here. Yes, the glucose, galactose, and fructose. Now, where they will go? They will go into the liver. Okay, they will go into the liver, right? Let us just try to draw the liver over here. Now, how they reach the liver, guys, you already know we have a very good portal venous system. We have a very good portal venous system. And through this portal vein, the glucose, galactose, and fructose will get absorbed. It will get absorbed. It will go into the liver. It will go into the liver, right? Now, over here, I am only talking about glucose now. Galactose and fructose, I will be talking about it in the further classes, the metabolism of galactose and fructose in the upcoming class. But over here, remember, beta, the glucose, it is going inside the liver, right? So over here, our favorite glucose has reached, right? Now this glucose, the excess glucose, okay, will be now converted into glycogen. It will be now converted into glycogen. Glycogen, yesterday we have discussed, it is basically a polymer of glucose only, glucose. They are joining together to form glycogen. Glycogen, it is the storage form of carbohydrates in humans. Obviously, excess amount of glucose, it is stored as glycogen in our body. So this process of formation of glycogen from the glucose, it is called as glycogenesis. It is formed as glycogenesis. Now, again, the name justifies, guys. Glyco means glycogen. Genesis means formation of glycogen from glucose. Now, suppose let us assume a condition. Let us assume a condition, guys, where you are fasting, right? What will happen? The glucose that is present in your body will automatically, okay, uh, in the initial stages, whatever the glucose is there, it will be getting used up. Later on, the glycogen will be broken down to release because every time you're not eating, right? So after some time, the blood glucose level will decrease. So automatically, this glycogen will be broken down to form again glucose, and this is called as glycogenolysis. It is called as glycogenolysis. So I will be discussing these two procedures, guys, the glycogenesis and glycogenolysis tomorrow, right? So again, you know where it is no starting from. We are consuming disaccharides. They are broken down into glucose. Glucose is entering into the liver. Glucose is forming glycogen. And in the fasting state, the glycogen is again broken down to form glucose. So we have two processes that we have to discuss, glycogenesis and glycogenolysis. Lysis means breakdown of glycogen to form glucose, right? Now, apart from that, let us focus that this glue okay now okay now this glucose will now enter into the bloodstream. It will now enter into the bloodstream. It will now enter into the blood. Okay, so this glucose will enter into the blood, and this blood, okay, will take the glucose to our very favorite cell. Okay, we have a cell. You already know cell, it is bounded by the cell membrane. So on the cell membrane, guys, so this is a cell and this is cell membrane, right? On the cell membrane, guys, we have certain transporters, right? We have certain transporters, and these transporters are called as GLUT. These are called as glucose transporters, right? These are called as glucose transporters, right? So obviously, this glucose which is present outside, right? The glucose which is present outside will be now entering into the cell. It will be now entering into the cell via blood. So bloodstream में glucose आया और blood से कहाँ जाएगा? Cell. Means in every part of the body, right? Whether it is pancreas or any part of the body, the brain, heart, everywhere, right? This glucose will now move into the cell with the help of a transporter that is situated on the plasma membrane that is called as the glucose transporter, right? So I hope till now it is clear. Yes. So now just try to see what happens inside the cell, guys. What happens inside the cell? Let us just assume that this is the cell membrane, guys. Now this is something very important, right? This is the cell membrane, guys. This is the cell membrane, right? Now on this cell membrane, we have, yes, we have this glucose transporters, right? We have this glucose transporters. And through these glucose transporters, what will happen? The glucose that is present outside will be now permeable to enter into the cell. Okay, this glucose will now enter into the cell. So I'm just drawing a cell in like this only. So let us suppose that this is the cell, right? So now this glucose will come into the cytoplasm of the cell. Okay, now the glucose has reached the cytoplasm, the jelly-like liquid of the cell, that is the cytoplasm of the cell, right? Now this glucose, now this glucose, what will happen in the cytoplasm? Now guys, again, very important. You guys already know, let us suppose that we have one molecule of glucose, 1 mole of glucose is there, and you already know the formula of glucose, C6H12O6, right? So we have 6 carbons in, yeah, we have already discussed aldehyde derivative yesterday. Okay, so this glucose in the presence of oxygen, will convert into pyruvate, and this is something which is called as aerobic glycolysis, right? This is called as aerobic glycolysis, which is in presence of oxygen, So I will be discussing how the glucose, it is getting converted into the pyruvate, right? So from one glucose, 2 pyruvates are formed. Again, pyruvate, it is a 3 carbon compound. Glucose, it is a 6 carbon compound. तो एक 6 कार्बन ग्लूकोज से 2 पायरुवेट बनते हैं क्योंकि पायरुवेट 3 कार्बन का होता है, ठीक है? सो ऑटोमेटिकली नो कार्बन इज लॉस्ट. अगेन mark my statement, no carbon is lost in the conversion from glucose to pyruvate. Carbon number remains the same. From one glucose, 2 pyruvates are formed. 3 कार्बन के 2 बन जाते हैं क्योंकि ग्लूकोज के पास 6 कार्बन है, right? Now this is something which is happening inside the cytoplasm. Obviously cell के अंदर सबसे पहले क्या आ जाता है? Cytoplasm. So if I ask you guys, the aerobic glycolysis basically takes place in cytoplasm of the cell, right? What is the site? It is the cytoplasm of the cell. So this is a cell. We have the cell membrane. So glucose is getting converted into pyruvate, and this is something called as aerobic glycolysis, which I will be discussing in the upcoming slides. So this is something called as aerobic glycolysis, the first pathway that we have to discuss today. The other one, guys, you already know. We have something. Okay, Okay, if oxygen is not there, right? In absence of oxygen, okay, in absence of oxygen, it will not form pyruvate, it will form lactate. Okay, it will form lactate. And this is called as anaerobic glycolysis. Okay, this is called as, yes, from glucose, what is formed? Lactate. This is called as anaerobic glycolysis. Anaerobic glycolysis. Now, anaerobic glycolysis, it is also occurring in the cytoplasm of the cell. Okay, now, we already know we have a cell organelle. Okay, we have a cell organelle that is present inside the cell, right? And this cell organelle is nothing but the mitochondria. It is nothing but the mitochondria. Now you already know, guys, the mitochondria, it is a double membranous structure. Okay, it is a double membranous structure. We have the outer membrane and we have the inner membrane of the mitochondria. So I'm not drawing the entire mitochondria. Now what will happen to this pyruvate, guys? This pyruvate will now enter into the mitochondria of the cell. Now this pyruvate will try to enter into the mitochondria of the cell. Now this pyruvate has entered into the mitochondria of the cell, and you already know pyruvate is a 3-carbon compound, right? Now what will happen? This pyruvate will form, yes, this pyruvate will form acetyl coenzyme A. I will be coming to this reaction. This reaction from pyruvate to acetyl coenzyme A, this is called as link reaction. I will tell you why it is called as link reaction, because it is linking the aerobic glycolysis to the TCA cycle. I will be talking about that. So this pyruvate to acetyl coenzyme A, this is called as the link reaction, and where this link reaction is happening in the mitochondrial matrix. Very, very important. If you draw it, it is very easier to remember. So it is happening in the mitochondrial matrix, okay? Not the outer mitochondrial membrane, not the inner, just inside the mitochondria. That is the mitochondrial matrix. This pyruvate is getting converted to acetyl coenzyme A. And remember, guys, the acetyl coenzyme A, it is a 2-carbon compound. So from pyruvate, from a 3-carbon compound, a 2-carbon compound is formed, which clearly indicates that a CO2 is lost, right? A CO2 is lost, okay, in the conversion of pyruvate to acetyl coenzyme A, right? That is again with the help of an enzyme that is called as pyruvate dehydrogenase. I will be teaching you all the enzymes, so no need to worry. So remember, this is called as pyruvate dehydrogenase. Pyruvate dehydrogenase. Now, in the mitochondrial matrix, let me just try to draw the entire mitochondria. Yes, this is again the entire mitochondria that I'm trying to draw in front of you. Now, this acetyl coenzyme A, in the mitochondrial matrix, guys, we have something called as oxaloacetate. Oxaloacetate, which is a 4-carbon compound. Now, acetyl coenzyme A will combine with the oxaloacetate to form something which is called as the citrate. Citrate. So citrate will again be a 6-carbon compound, because acetyl coenzyme A 2-carbon, oxaloacetate, which is present in the mitochondrial matrix, it is a 4-carbon, 4 plus 2, 6. And what is the name of the compound? Yes, it is citrate. Now citrate, again, it will go a series of reaction and it will form alpha ketoglutarate. I will teach you the entire cycle, no need to worry. Alpha ketoglutarate, and remember, it is a 5-carbon compound, right? Again, a CO2 molecule is lost over here from conversion of 6-carbon to 5-carbon. This alpha ketoglutarate, okay, will form succinyl coenzyme A, right? Succinyl coenzyme A again is a 4-carbon compound, so obviously a CO2 molecule is lost. And this succinyl coenzyme A, again, a series of reaction will take place and it will form oxaloacetate. So this cycle from citrate to alpha ketoglutarate, from succinyl coenzyme A to oxaloacetate, this entire cycle, it is called as citric acid cycle, or it is called as Krebs cycle. It is called as Krebs cycle, or it is also called as the citric acid cycle. Okay. So again, guys, what all points that we have to discuss, I have just given a very, in a very short form, I have given you the entire flow chart. कि कहाँ से start हुआ है. We have taken the disaccharide going into the stomach, converting into glucose. Now this glucose is going to the liver, okay, where it is forming glycogen forming glucose. Again, the process of reactions. 2 reactions, I will be discussing it tomorrow. From the liver, it goes into the blood. From the blood, it goes into the cell. In the cytoplasm, the glucose will convert into pyruvate, and this is called as aerobic glycolysis, which we are going to discuss. If oxygen is not there, okay, then what it will form? Lactate, that is called as anaerobic glycolysis. Two cycles, aerobic and anaerobic, both are occurring in the cytoplasm. Now, this pyruvate will try to enter the mitochondria. It will go into the mitochondrial matrix, right? And it will form acetyl coenzyme A. Now, again, why I said link reaction? Because this pyruvate to acetyl coenzyme A, basically this pyruvate is coming from aerobic glycolysis, and this acetyl coenzyme A, which is formed, it is undergoing Krebs cycle. So this link reaction, it is basically connecting the aerobic glycolysis with the Krebs cycle. So this acetyl coenzyme A will react with oxaloacetate to form citrate, alpha ketoglutarate, succinyl coenzyme A. This entire cycle, it is called as the Krebs cycle or the citric acid cycle. The Krebs cycle or the citric acid cycle. So that is something we have to discuss. Krebs cycle also, aerobic glycolysis also, anaerobic glycolysis also. Right? So that is the three processes that we have discussed: aerobic, anaerobic, and citric acid cycle. And over here we have discussed glycogenesis and glycogenolysis. So at least mota moti, I have already told you the 5 main procedures in the carbohydrate metabolism. Right? So now, without wasting much more time, guys, let us just start with the glycolysis wala part. Let us just start with the glycolysis wala part. So now now guys, I hope this is you understood, guys, the entire overview of the carbohydrate metabolism. Just giving a thumbs up in the chat box. Sarath, if you are there, right, just let me know. Okay, so now let us start with the aerobic glycolysis first. Okay, the glycolysis, what is the concept, guys? You already know. Glyco means glycics, right? The glycis means beta sweet sugar. Okay, it means sweet sugar, or I can simply say the glucose wala part, and lysis means, you already know, breakdown. It means breakdown. So basically the sweet sugar or the glucose, it is broken down to form or to liberate energy, right? To liberate energy in the form of ATP, that is the energy currency of the cell. That is the energy currency of the cell. Now, this glycolysis, guys, okay, 2-3 important points from the exam point of view that you have to remember, that this occurs in all organs of our body. This glycolysis occurs in all organs of our body. Secondly, it occurs in the well-fed state. It occurs in the well-fed state. Now, well-fed state is that state, it is usually 1 to 4 hours after food. It is usually 1 to 4 hours after food. जब आपने बहुत अच्छे से खाना पीना खाया हुआ है, उसी टाइम पे ग्लाइकोलिसिस होगी. Whenever you take glucose, the glucose will go inside all the cells of the body and they will undergo a series of reaction to produce ATP, right? So obviously, now, again, under influence of which hormone the glycolysis takes place? Again, guys, you already know that, for example, if I drink 1 liter of Coca-Cola right now, what will happen? Again, the glucose level will increase in the blood. Okay. अगर blood में glucose level बढ़ गया, so obviously it will provide an obstruction to the flow of the blood. And this is the cell, right? Cell has a limited capacity to not take glucose. Suppose 100 molecules are there in the blood stream. Okay, in the blood, 100 molecules of glucose are there, and a cell has a capacity of taking entry of only 50 molecules. I'm just taking a hypothetical example, guys. So if 100 molecules of glucose are there and cell has a capacity of taking only 50 molecules, guys, now again the 50 molecules are there in the blood. They will provide an obstruction. And if the blood is containing glucose, it is obviously known as the diabetic state, right? So after drinking 1 liter of Coca-Cola, every person in the world should have diabetes. No, it's not like that. So what is the extra thing that is happening? You already know we have something called as the pancreas, guys, right? We have something called as the pancreas, and pancreas, we have the beta cells which releases insulin. Now, this insulin will be released in case of excessive glucose in the blood. अगर blood में extra 50 glucose पड़े हुए हैं, what will happen? This will again give a signal to the beta cells of the pancreas to release insulin. This insulin will go and it will again force the cell to take extra 50 glucose so that there is no glucose left in the blood. ब्लड के अंदर ग्लूकोज नहीं रहना चाहिए. Glucose should go inside the cell. Again, it is a safe condition. If it is there in the blood, the patient is diabetic, right? Again, one situation can happen, no, if the patient agar insulin hi nahi ban raha hai, right? Or there is less production of insulin, diabetes, right? So that is something that you have to remember, that glycolysis takes place in, okay, under level of which hormone? That is insulin. Okay, under influence of insulin, the glycolysis takes place. Glycolysis takes place. Now, this glycolysis can occur aerobically also in the presence of oxygen, and anaerobically also in the absence of oxygen. Okay. It can occur in the presence of oxygen, that is aerobic glycolysis, and in the absence of oxygen, that is anaerobic glycolysis, right? So now, again, one important concept that I want to teach you guys, right? Okay. Again, okay. Okay. Before coming on to the site and organelle part, okay, let us just discuss one more concept over here. That is something important. I wanted to discuss this. Yes, that is the erythrocyte, guys. You already know the erythrocyte has no nucleus. The RBCs, guys, the red blood corpus cells have no nucleus. The adult RBC has no nucleus. They have no mitochondria. They have no mitochondria. Obviously, for glycolysis, either oxygen should be there or right. Now, the erythrocytes, they depend, because erythrocyte may तो mitochondria है ही नहीं, so obviously pyruvate will not be able to go further to continue the TCA cycle or something that's not present. But so this erythrocyte, it is solely responsible for this aerobic glycolysis. So this erythrocyte, the RBCs, depends only on glucose, guys. Very, very important. It depends only on glucose, right? Because it has no mitochondria. So there is no TCA cycle which is occurring. So it, if there is any deficiency of glycolytic enzymes, very, very important. Again, a 1 marker question which is being asked in the practical or Viva exam. If there is deficiency of glycolytic enzymes, guys, glycolytic enzymes, remember, it can lead to defective hemolysis. Hemolysis means breakdown of the RBC, right? Okay, the membrane of the RBC will rupture and the hemoglobin will come out. Hemolysis, right? Again, one more important clinical aspect that I wanted to discuss with you guys, that is regarding the skeletal muscle. That is regarding the skeletal muscle, guys. Now, skeletal muscle is basically having low blood flow. Okay. Now, skeletal muscle having low blood flow in cases of exercise, guys. Whenever you go to gym, guys, right? Whenever you go to gym, what will happen if you do continuous exercise? What will happen? There will be low blood flow, means low oxygen. So automatically, if there is low blood flow, basically the oxygen going to the muscles will be less, right? Okay, that is something called as ischemia. Ischemia, that is low blood flow or less blood supply. Okay, blood supply कम होगा तो oxygen supply भी कम हो जाएगा, right? So in that case, if the oxygen is not going properly, will the skeletal muscles die? No, no. The skeletal muscles can survive ischemia, right? They can survive ischemia. Okay, they have a possibility of surviving ischemia, but only on one condition, if the glycolytic enzymes are present. Now imagine a condition where the glycolytic enzymes are absent. Glycolytic enzymes are absent. What will happen if there is deficiency of glycolytic enzymes? This skeletal muscle will become fatigue. This skeletal muscle will become fatigue, right? The skeletal muscle will become, that is why the glycolysis is very, very important, no, in cases of RBCs, also in cases of skeletal muscles as well. So that is regarding the concept. Coming on to the site and organelle of aerobic glycolysis. Remember guys that the site, it is all organs, the organelles, right? It is the cytoplasm of the cell. Even I don't have to tell you, in the overview, it is very, very clear. The concept I have told you with respect to erythrocytes and skeletal muscle, why it is needed, right? Now coming on to the steps. Now, guys, according to the notes that are given in the Auricle, they have beautifully curated the notes firstly. So they have already mentioned all the steps. I will just try to summarize what they have mentioned in the notes. And guys, later on, you can just check the notes on the app, how it will know. Now, there are two phases, guys. And believe me, if you write the answer that is given, just like I'm drawing, I'm also copying from the flowchart that is being given in the notes, right? So there are two phases, guys. Firstly, it is called as the preparatory phase. It is called as the preparatory phase. The other phase, it is called as the payoff phase. The payoff phase. The preparatory phase, it is the phase which we are preparing the glucose. Now, preparation. We are preparing glucose for making more ATP, right? We are preparing the glucose for making more ATP, right? Now, when you're preparing someone, you have to put your energy to prepare someone. So obviously in this phase, remember guys, the ATP is used. The ATP is used. And this preparatory phase, it is again divided into two parts. The first part, it is stage of phosphorylation. Phosphorylation. I will be talking about this stage of phosphorylation in detail. No need to worry. Phosphorylation का मतलब होता है to add the phosphate group. To add the phosphate group, right? To add the phosphate group. And the second one, it is stage of splitting. Stage of splitting, right? We have stage of phosphorylation and we have stage of splitting. Now, coming on to the payoff phase, पे का मतलब क्या होता है? ATP generate होगी. So ATP is generated in this phase. ATP is generated in this phase. Basically, over here, both the process will occur, oxidation and reduction. Both will occur. Oxidation and reduction both will occur. So we have stage of phosphorylation, stage of splitting, and payoff phase. Payoff phase. So in some books, it's like the preparatory phase is divided into two, or they have simply written stage of phosphorylation, stage of splitting, and payoff phase. So let us start with the very first one, that is the stage of phosphorylation. Stage of phosphorylation. Stage of phosphorylation. Now guys, you already know phosphorylation, basically we are adding phosphate. We are adding phosphate group. Now, obviously we have to start with the glucose. Try to be very clear guys. I will be teaching you once and it is very, very, very simple. Don't try to ratify anything. Just try to understand the cycle very clearly over here. Now glucose guys, it is made up of 6 carbon molecules. Okay, it is made up of 6 carbon molecules, right? We have 1, 2, 3, 4, 5, and 6. Now this glucose, you already know it is the formula is C6H12O6. It is made up of 6 carbon molecules. Now, what we are doing in the stage of phosphorylation, guys, we are adding phosphate. We are adding phosphate. Okay, phosphate आएगा कहाँ? मैंने अभी तुमको लोग को बताया the ATP is used in this preparatory phase. So obviously we have something called as the adenosine triphosphate, which will be broken down into adenosine diphosphate. Okay, ATP will convert into ADP. So triphosphate convert into diphosphate. So automatically one phosphate molecule is generated in this phase. Now it will go to the 6th carbon atom of the glucose and it will attach there. So again, this is called as glucose 6 phosphate. Glucose 6 phosphate. Now what is glucose 6 phosphate, guys? This is nothing. You already know we have the 6 carbon atoms. Okay, we have the 6 carbon atoms, and to the 6th carbon atom, what is attached? The phosphate molecule is attached. This phosphate, it is coming from ATP. ATP gets converted into ADP, and what is released? This phosphate, and this phosphate, it comes on to the 6th position. That's why it is glucose 6 phosphate. 1, 2, 3, 4, 5, 6. Now, ATP is getting converted to ADP. It is with the help of enzyme. You already know it is called as hexokinase or glucokinase. I will be coming to the difference. Just remember right now we have hexokinase or glucokinase, right? And this conversion, again, a very important question from the exam point of view, guys. You should remember an ion is required that is called as magnesium ion. Magnesium ion. Now, when we say kinase, guys, whenever you see the word kinase, again, try to build the concept, guys. Whenever you say kinase, guys, you should remember it is the transfer of phosphate group. Okay, it is the transfer of phosphate group from one molecule to another. From one molecule to another. Now, guys, you already know. Now, which one molecule is there? ATP. And which is the other molecule over here? Glucose. So the phosphate, basically the ATP is losing one phosphate, and that phosphate it is going to the glucose. And which carbon atom of glucose? The 6th carbon atom. So anywhere the kinase is used, you should remember it is the transfer of phosphate group from one component to another component, right? From one molecule to another. Now from glucose 6 phosphate, now from the glucose 6 phosphate, guys, remember from the glucose 6 phosphate, okay, there is something which is formed as fructose 6 phosphate. We have fructose 6 phosphate. From glucose 6 phosphate, we have fructose 6 phosphate, right? Now, guys, glucose is also containing 6 carbon. Fructose is also containing 6 carbon. Again, glucose is aldehyde derivative. Fructose is keto derivative, right? Now, if we have the same number of carbon atoms, okay, we have the same number of carbon atoms. We have the same number of carbon atoms, and the phosphate is present again at the 6th carbon atom. Now, this is something called as the isomers, right? Agar same number of carbon atoms hote hai, but the structure is different. Glucose is a different structure because it has aldehyde group. Fructose is a different structure because it has keto group. So from glucose 6 phosphate, fructose 6 phosphate is formed. And the enzyme that will be used over here, it is called as isomerase. Isomerase. And okay, what will be the name of the isomerase? Phospho hexose isomerase. Again, very easy. Phospho because the phosphate group is attached over here. Hexose, why? Because there are 6 carbon compounds and isomerase is this, right? Now, that is but the structure is different. Glucose is different, fructose is different. Now, again, there are two important things, guys, that you have to focus over here, right? The two important things that you have to focus over here. The glucose 6-phosphate can also enter HMP pathway, right? There is hexose monophosphate pathway, or it can also undergo uronic pathway. I will be discussing it later on. Okay, or it can also undergo glycogen synthesis. Glycogen synthesis. So there are many fates of glucose 6-phosphate. It can go either ways. It can go to HMP pathway, it can go to uronic pathway, it can go glycogen synthesis. It can, but when it forms fructose 6-phosphate, now this is something called as the bottleneck of the pathway. Means अब तो ये आके यहाँ फंस गया. It cannot be no. Yeah. Now, whenever we use the term isomerase, guys, you just have to remember that it is a reversible process, right? It is a reversible process, right? So, yes, glucose is basically getting committed. The glucose is getting committed for the cellular pathway. Now, this is something remember, it is the reversible step. Okay, this is the reversible step, right? This is the reversible step. Now, I will be continuing on the next slide. Now this fructose 6-phosphate, okay, let me just try to write it over here only. This fructose 6-phosphate will now form fructose 1,6 biphosphate. Now this is something which is called a stage of phosphorylation. Now you guys already know fructose 6-phosphate converting into fructose 1,6 biphosphate. So it clearly means, guys, that if we have the 6 carbon compounds, right, on 6th place we already have phosphate. Now at the first carbon atom also we have phosphate. So we have 1, 2, 3, 4, 5, 6. So first place also and 6th place also. Now, obviously, guys, again, we are adding one more phosphate. You guys are very smart, so you should know that over here, we are again using the ATP. We are again using the ATP to form ADP, right? Again, which enzyme will be used, guys? Yes, very good. That is the kinase enzyme. Because we are shifting one phosphate from ATP to fructose, right? So that is something which is called as phosphofructokinase. Phosphofructokinase. Obviously, we have the phosphate group. The fructose is getting converted, right? So it is phosphofructokinase. Wherever we are using kinase, it means we are shifting one ATP from one molecule to another. We are shifting one ATP from one molecule to another, right? So that is something you guys have to know. That is the stage of phosphorylation. Glucose 6-phosphate, phosphate ऐड हो गया. Glucose 6-phosphate आइसोमर करो, उसका फ्रक्टोज बन जाएगा. और फ्रक्टोज 6-phosphate में फिर you are adding what? Phosphate, fructose 1,6 biphosphate. So that is something you guys have to remember, right? Now coming on to yeah. So we have discussed the stage of phosphorylation. We have discussed the stage of phosphorylation. Now coming on to the stage of splitting. The second one, it is the stage of splitting wala part. Stage of splitting. The stage of splitting. Now, now guys, you already know from where we left. We left at fructose 1,6 biphosphate. Right? इसका मतलब कुछ नहीं. We have 6 carbon atoms. 6th carbon atom is having phosphate. First carbon atom is also having phosphate. This is fructose 1,6 biphosphate. Now this will get split into two components, splitting. Okay, it will get into split into two components. Now, how the splitting takes place, guys? 3 carbon going to one side, 3 carbon going to one side. And you already know the two carbohydrate that we formed yesterday from 3 carbons from glycerol. Basically, two carbohydrates are formed. Yesterday only we discussed. We have dihydroxyacetone, which is having the 3 carbon compound, right? And we have glyceraldehyde, right? So one, we have 3 carbon compound, right? The dihydroxyacetone and glyceraldehyde, right? Now, dihydroxyacetone also 3 carbon with a phosphate, glyceraldehyde 3 carbon with a phosphate. Now, that is something called as the, and we have already discussed the dihydroxyacetone and the glyceraldehyde yesterday, how they are formed from the glycerol part. Over here, they are also getting the fructose is getting splitted into these two parts, right? Now, guys, this dihydroxyacetone and glyceraldehyde, again, you can see something very common. They both are containing 3 carbons, just like glucose and fructose. So obviously, they can change either ways, right? They can change into each other, right? These are isomers. And yes, this is the enzyme which is used over here, that is phosphotriosis. Triosis means they are having 3 carbons, isomerases, right? Isomerase. If simply you remember isomerase also, it's same. Now, this dihydroxyacetone phosphate is getting converted into 2-glyceraldehyde, okay? So I will be talking about glyceraldehyde only. So basically we have 2 molecules of glyceraldehyde now over here, okay? Because dihydroxyacetone, it is also converting into glyceraldehyde. It is also getting converted into glyceraldehyde. And how many glyceraldehyde are there? 2, because the dihydroxy is also getting converted into glyceraldehyde. So we have 2 glyceraldehyde. One was automatically there, and dihydroxy also get converted into glyceraldehyde. Now, this 2-glyceraldehyde, okay, this 2-glyceraldehyde. Now we come on to the payoff phase, guys, the last phase that we have discussed, right? The payoff phase. The payoff phase. These are all from the notes of the Auricle, right? The payoff phase, guys. Now this 2-glyceraldehyde. Now, so I just wanted to name it completely, dihydroxyacetone phosphate, because phosphate group is attached, and glyceraldehyde. Which carbon atom is containing phosphate? The third carbon atom. So it is glyceraldehyde 3-phosphate. Okay, it is glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate. Now, this glyceraldehyde 3-phosphate will now convert into 1,3-bisphosphoglycerate. Will now convert into 1,3-bisphosphoglycerate. It will now convert into 1,3-bisphosphoglycerate. Now, this is something where the substrate level phosphorylation is basically taking place. What will happen, guys? Over here, the NAD. NAD basically stands for nicotinamide adenine dinucleotide, right? This NAD plus, okay, will change into NADH plus H plus. I will be coming to the concept of oxidation and reduction. I will teach you, right? And this is basically taking place with the help of an enzyme that is called as glyceraldehyde 3-phosphate dehydrogenase, right? Dehydrogenase. Now, dehydrogenase is basically a group of enzyme oxyreductases. Okay, oxyreductases. Now, what this dehydrogenase do, guys? What is this dehydrogenase do? Okay, it will take. Okay, let us just simply. I want to make you understand. If there is one molecule which is containing H2 and the other molecule which is not containing H2, what will happen? This A will give H2 to B. So A will be left alone and B will contain now H2. So basically the hydrogen is getting transferred from one substrate to another. That is a very simple reaction where a dehydrogenase can be explained. Dehydrogenase. So obviously you can see NAD plus is basically losing H2 so that it is no getting the other one getting converted into NADH, right? Now, 1,3-bisphosphoglycerate. 1,3-bisphosphoglycerate. Obviously, guys, one phosphate molecule is basically added over here, right? So 1,3. So you already know we have 3 carbon. So on the third carbon also the phosphate is added, and all on the first carbon also the phosphate is added. 1,3-bis. Bis means bi means 2. 2 phospho in the glycerate, the glyceraldehyde component, right? Now, how many molecules of 1,3-bisphosphoglycerate will be formed? It is 2. It is 2. 2 glyceraldehyde, 2 1,3-bisphosphoglycerate. Now, from 1,3-bisphosphoglycerate, what will happen? Yes, very importantly, it will be forming 3-phosphoglycerate. 3-phosphoglycerate. Now, this phosphate, guys, if you ask me, this is an inorganic phosphate. It's not coming from the ATP or something, okay? It is coming from somewhere else, right? So this is an inorganic phosphate, right? So over here, from 1,3-bisphosphoglycerate, only 3-phosphoglycerate is formed. So means one phosphate is lost. So it is again used by the ATP. Okay, it is again used by the ADP, adenosine diphosphate, to form ATP. Okay. Okay, this phosphate from 1,3-bisphosphoglycerate to 3-phosphoglycerate automatically means that one phosphate is lost. One phosphate, it is going to ADP. Okay, and phosphate is getting attached to ATP to form ATP. ATP, right? Now this is something. Now, this is something which is, you already know, movement of phosphate from one molecule to another. It is called as kinase, right? It is called as kinase. And this is called as phosphoglucokinase. Phosphoglucokinase. Kinase का मतलब क्या होता है? Phosphate is going from one molecule to another. Phosphate is going from 1,3-bisphosphoglycerate to ATP. ATP is another molecule. 1,3-bisphosphate is another molecule. 1,3-bisphosphate is giving phosphate to ATP so that ATP gets converted into ATP. Adenosine diphosphate will get now converted into adenosine triphosphate. Now this 3-phosphoglycerate, will be now converted into 2-phosphoglycerate. 2-phosphoglycerate. You guys already know we have the 3 carbon atoms, right? To the third carbon, the phosphate is attached. 2-phosphoglycerate basically means the phosphate is basically shifted to the second carbon, 1, 2, and 3. In a same compound, if the phosphate is shifting from one carbon to another carbon, now there are not 2 different molecules. The molecule is same, the phosphoglycerate. But the phosphate, which was earlier present at the third carbon, is now present on the second carbon, is again taking place with the help of an enzyme that is called as mutase. It is called as mutase. Mutase means basically it is the transfer of phosphate from within the same molecule. Okay, within the same molecule, the phosphate, which was earlier present at one carbon, it is now present at another carbon. Earlier it was present at the third carbon, it is now present at second carbon. The component is same. That's why mutase. If the components are different and phosphate is transferring from one molecule to different molecules, it is kinase, right? If the molecule is same. So obviously the only the phosphate positioning is changing. It is mutase, right? Now this 2-phosphoglycerate, this 2-phosphoglycerate will now get converted into phosphoenol pyruvate. It will now get converted into phosphoenol pyruvate. It will now get converted into phosphoenol pyruvate. Now, this reaction basically takes place with the help of a loss of water molecule, right? Now, now, so yes, phosphoenol pyruvate, right? And this phosphoenol pyruvate will further change into, okay, conversion of phosphoglycerate to phosphoenol pyruvate will take place with the help of enzyme enolase, right? And later on, guys, just remember phosphoenol pyruvate will change into pyruvate, right? The phosphoenol pyruvate will change into pyruvate. Basically, over here, again, the phosphate group is getting lost. So ADP will use this phosphate, right? The phosphoenol while apart, and it will convert into ATP. It will convert into ATP. This process, it is called as substrate level phosphorylation. It is called as substrate level phosphorylation. Phosphorylation. Okay, it is called as substrate level phosphorylation. So you can very well see in the two slides I've already shown you guys from glucose how the pyruvate is formed. From glucose, how the pyruvate is formed. From glucose, how the pyruvate is formed, right? So that is something you have to remember. Now, if you are done with this, guys, stage of phosphorylation, stage of splitting, and the payoff phase, it is very easier to know. Firstly, discuss the. Okay, let us first discuss the. Okay, first discuss the energetics, guys. Very, very important. We have already discussed this, so I'll be just going very fast in this particular slide. So you already know glyceraldehyde 3-phosphate dehydrogenase. Now coming on to the cycle, guys. Cycle में देखो. यहाँ पे मैंने पढ़ाया. I will be dealing it with the green color. Glyceraldehyde 3-phosphate dehydrogenase. NAD is getting converted into NADH plus H plus. So you should remember, guys, that one NADH, it is giving 2.5 ATP, and 2 NADH will give 2.5 into 2. How many ATP? 5 ATP. That is coming on payoff phase में ध्यान देना है. Obviously, in the starting, the ATP is getting utilized. Splitting reaction, no need of ATP in the starting, no need. Come to the payoff phase where actually you're getting the ATP. So 2 NADH will give 2.5 into 2, that is 5 ATP. Coming on to the phosphoglycerokinase. Okay. The phosphoglycerokinase, guys, you already know, we have already discussed this, right? The okay, glucokinase, you already know, the ATP is generated over here. Obviously, ये 2 molecules है ना guys, 2 molecules से मैंने बताया था. So ये 2 into this ATP. Yes, so we have 2 ATP over here, 5 from that. Obviously, वो 2 molecules है ना, एक NADH से 2.5, but we have 2 glyceraldehyde 3-phosphate. So it is 5. And pyruvate kinase at the last, you already know. Yes, pyruvate kinase, I have already told you 1 ATP is formed. Okay, the substrate level phosphorylation. Over here, the enzyme that is used, that is pyruvate kinase. Again, kinase, guys. Kinase, you all remember now, kinase is transfer of phosphate from one molecule to another. The ATP is one molecule, phosphoenol pyruvate is one molecule. So from phosphoenol pyruvate, this phosphate group is going to ADP to convert into ATP. So phosphoenol pyruvate is another, ATP is another. So automatically one, I mean the two, obviously दो हो गए ना. So we have 2. So total ATP, basically it is 5 plus 2 plus 2, that is 9 ATP. But again, guys, if I ask you the net gain of the ATP, okay, if I simply ask you the net gain is not 9 ATP, guys. Net gain is because in the starting, guys, you already know, yes, you already are aware of the fact that 1 ATP is lost in the very first reaction. and 1 ATP is also lost in this third reaction. So 2 ATPs are lost. The very first one is the hexokinase wala part, and the second one it is the phosphofructokinase wala part. So basically 2 ATP molecules are lost in the very preparatory phase, right? The preparatory phase, right? That is the stage of phosphorylation. So 2 ATPs are automatically lost in the first part. Hexokinase, 1 ATP lost. Phosphofructokinase, 1 ATP lost. So 9 minus 2, how much is the net gain of the ATP? That is 7 ATP. The net gain of the ATP, it is 7 ATP. So that is simply about the energetics of the this glycolysis. Simply the energetics of this glycolysis. Coming on to the irreversible reaction, guys, there are total 10 reactions of this glycolysis. 3 are irreversible. Now, the very first one, hexokinase and glucose kinase wala reaction, the first one, right? Where the glucose is getting converted into glucose 6 phosphate, that is an irreversible reaction. The third reaction, guys, you already know. The third reaction, guys, you are very much aware when the fructose 6 phosphate is getting converted into fructose 1,6 biphosphate. Again, this is an irreversible reaction, right? And the last reaction, the pyruvate kinase wala reaction, where the ATP is formed, right? The pyruvate kinase wala reaction, where the phosphoenol pyruvate changing into pyruvate, it is again irreversible. So just remember the irreversible one, the rest all are reversible reactions. So we have 3 irreversible reaction and 7 reversible reactions. Now coming on to the clinical correlations over here, very, very important, the clinical correlations. Guys, you should understand that pyruvate kinase deficiency, okay, it is the second most enzyme deficiency in humans, and it can lead to hemolysis. I already told you in the starting that the RBC is solely responsible for this aerobic glycolysis wala part. Now, if suppose the glycolytic enzyme, the pyruvate kinase, the phosphate wala part is already absent, then how the ATP will be formed? The RBC will not be able to survive, and RBC will burst out. So all the hemoglobin will come out. So that is the hemolysis, right? The breakdown. Secondly, the phosphofructokinase deficiency. I've already told you in the starting the concept of the skeletal muscles. Now, if it is deficient, guys, what will happen? The skeletal muscle fatigueness will take place. This is a very important part of the glycogen storage disorder. This is very, very important part of the glycogen storage disorder. So that is something you guys have to remember. Now, so I hope this is clear, the clinical correlation, the energetics, the irreversible reactions of glycolysis. And again, a very important 1 mark question that is being asked, that what is the rate limiting enzyme of this glycolysis? That is phosphofructokinase 1. It is phosphofructokinase 1. That is the rate limiting enzyme, right? Inhibitors again are very, very important. Now we have something called as enolase, right? Where the water molecule was removed. So you should remember there is a component called as fluoride, which inhibits the enolase. Okay. Iodoacetate inhibits glyceraldehyde 3-phosphate dehydrogenases. Okay. The payoff phase, the very first reaction of the payoff phase, right? And arsenate basically inhibits 1,3-bisphosphoglycerate kinase. 1,3-bisphosphoglycerate kinase. So these are the inhibitors of glycolysis: fluoride, iodoacetate, and arsenate. At what level they are inhibiting, that you have to remember, guys, right? Now, coming on to the regulation of glycolysis, the glycolysis is basically regulated by 2 pathways, guys. We have the hormonal regulation and we have the allosteric regulation. Hormonal regulation, you already know the glycolysis is basically taking place in the well-fed state, in the okay, in the under the influence of insulin, and it is activated in the dephosphorylated state. Obviously, अगर phosphorylation नहीं होगा तभी तो वो सारे activate होंगे ना, the hexokinase, the phosphofructokinase, and the pyruvate kinase. Obviously, these enzymes will be there only then, though the glycolysis will take place. The allosteric regulation, basically you already know from substrate what is formed product. So substrate always favors the forward reaction and product inhibits the forward reaction. So obviously if we talk about glucose is getting converted into glucose 6 phosphate, what is the substrate? Glucose. So glucose will be favoring the forward reaction. Glucose 6 phosphate will be an inhibitor. Okay, more amount of glucose 6 phosphate will be formed, it will automatically become a signal to stop कि नहीं और glucose 6 phosphate नहीं चाहिए. So always the substrate is basically favoring the reaction, okay, but the product it is always trying to inhibit कि we have sufficient amount of product, no need to further uh, there's no further need of glycolysis, right? So that is how it is maintained in our body, the hormonal regulation and the allosteric regulation, guys, right? So that is regarding the glycolysis wala part, right? That is again regarding the glycolysis wala part. So we have already discussed the overview of the carbohydrate metabolism, the glycolysis concept, site organelle steps, right? Clinical correlation, energetics, irreversible reaction, rate limiting enzyme, inhibitors, regulation. So every point related to glycolysis is covered. So if it is a 10-mark question, you have to write everything that is mentioned. Right, all the things. They can ask you a very short part also, how the regulation of glycolysis take place. So just mention this much. That is way enough for a 2-mark question, right? So that is how it is taking place. Now coming on to the anaerobic glycolysis, guys. Again, it is a very important topic. If you know glycolysis, this becomes very, very easy. So I hope you guys are not tired. Just give me a thumbs up, guys, if you are present, listening to me, right? Just give me a thumbs up that yes, Ishika, I can see. What about Sarath? Sarath, very good. I hope, Sarath, you are understanding. Ishika, you are understanding whatever we have discussed in the starting, all the steps that I have been discussing with you. If you guys have any doubt, Peter, please let me know there and then, right? Both of you. Coming on to the anaerobic glycolysis, guys, again, it is very, 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 very easy. Now, aerobic glycolysis basically beta takes place in the presence of oxygen and mitochondria. Anaerobic glycolysis, either oxygen should be there, either oxygen is absent or mitochondria is absent, right? Now, now, guys, you already know. Okay, let us start the steps, and you already know the steps. Glucose converting into glucose 6 phosphate, glucose 6 phosphate converting into fructose 6 phosphate, right? Glucose 6 phosphate converting, all the steps of the glycolysis, aerobic glycolysis, just come to that. Fructose 1,6 biphosphate getting converted, splitting reaction. Dihydroxyacetone, glyceraldehyde 3 phosphate. The initial wala jo part hai, this is the stage of phosphorylation. Stage of phosphorylation. This is splitting reaction. Again, it is same for aerobic glycolysis also. It is same for anaerobic glycolysis also. It is same for anaerobic glycolysis also. Now what is again change? What is the difference between aerobic and anaerobic? We'll try now to figure out over here. Right now, so let's continue from where we have left over here. Dihydroxyacetone phosphate getting converted into glyceraldehyde 3 phosphate. Now, dihydroxyacetone phosphate. Okay, basically the 3-carbon compound, it is getting changed into glyceraldehyde 3 phosphate. Okay. Now, how many glyceraldehyde 3 phosphate are formed? 2. 2 are formed, right? Now, what will happen, guys? This glyceraldehyde 3 phosphate will now change into, okay, 1,3 bisphosphoglycerate. Again, the same reaction. Bisphosphoglycerate. Okay, this glyceraldehyde 3-phosphate will now change into 1,3-bisphosphoglycerate. Now this 1,3-bisphosphoglycerate will again change into 3-phosphoglycerate. 3-phosphoglycerate will again change into 2-phosphoglycerate. Right? Now this 2-phosphoglycerate will change into phosphoenol pyruvate. And this phosphoenol pyruvate will now change into pyruvate. Right? This is something which we discussed, which we already discussed in the glycolysis. Again, it is the same. Now, what is the difference? If there is no mitochondria, like in RBC, we are telling this anaerobic glycolysis is taking place in the RBC. Now, in RBC, as we have already discussed the overview, guys, in the overview, we have already seen the pyruvate goes inside into the mitochondria, but RBC में तो mitochondria है नहीं. So what will happen now? This pyruvate will now convert into lactate, right? Because there is no mitochondria. So obviously, if the mitochondria is also absent, again, it will in turn undergo anaerobic glycolysis, right? So that is something that you have to remember, guys. Now this pyruvate will be changing into lactate, क्योंकि pyruvate को आगे जाने के लिए mitochondria है ही नहीं RBC में, तो automatically इसको lactate में change करना पड़ेगा with the help of enzyme lactate dehydrogenase. Now, how it is changing into lactate dehydrogenase, guys? You already know glyceraldehyde 3-phosphate is changing into 1,3-bisphosphoglycerate. So basically, it's a dehydrogenase, the NAD plus getting converted into NADH plus H plus. Now, what is happening, guys? This NADH plus, this NADH, which is used for providing 2.5 ATP, in turn 5 ATPs, because there are two molecules. एक NADH से 2.5, 2 से 5 हो जाता था. This NADH will not now no longer be able to provide ATP. It will come over here. Okay, it will be used by this lactate dehydrogenase, and this NADH H plus will be converted into NAD plus. So this is the entire cycle which is happening. Now, what is the difference between aerobic and anaerobic? Earlier, this NADH was giving ATP. Now it is not able to give any ATP right now. It is actually utilized in the last most step. Why pyruvate is converting into lactate? Because pyruvate cannot go into the mitochondria because there is no mitochondria in RBC. So it has to go into lactate. Overview clear है तो ये चीज बहुत clear हो जाएगी. So automatically 5 ATP has already gone. The 5 ATP has already gone, guys. So yes, the 5 ATP has already gone. So a question comes, what is the net gain in anaerobic glycolysis? Anaerobic glycolysis. So you already know we are getting 1 ATP from here. I mean 2 ATPs from here, right? We all are getting 2 ATPs from here because these are all 2, 2, 2, right? So ATP is converting into ATP. We are getting 2 ATPs from here. 2 ATPs, 2 ATPs from here, right? So basically, net gain in the anaerobic glycolysis, you already know 2 ATPs are utilized, right? In the starting phase, the 2 ATPs are utilized and automatically the 4 are formed. 2 plus 2, 4. Okay, so 2 ATPs are utilized. Okay, and total ATPs which are formed, these are 4, right? 2 from here, 2 from here. NADH will not be able to give ATP this time because it is used by this lactate dehydrogenase. So it's again a circle. It's not able to release energy, right? So what is the net gain, guys? The net gain of the ATP. The net gain of the ATP, it is 4 minus 2, that is 2. That is 2 ATPs. that is 2 ATPs, right? Aerobic glycolysis में 7 ATP का net gain था, anaerobic glycolysis में 2 ATPs का net gain है. You can easily understand the difference. You can easily understand the difference, right? You can easily understand the difference. So that is about the anaerobic glycolysis wala part. So we have completed aerobic glycolysis. We have also completed anaerobic glycolysis over here, right? So yeah. Now coming on to the next one, that is the link reaction. Very, very important. The link reaction. They will definitely ask you. So again, coming on to this particular overview, guys. Now we have already discussed. Just try to see my highlighter. We have already discussed the aerobic glycolysis. We have seen how the glucose is getting converted into pyruvate. We have also discussed the anaerobic glycolysis, how the glucose getting converted into lactate from pyruvate to lactate, right? Anaerobic glycolysis done, aerobic glycolysis done. Now this pyruvate, after aerobic glycolysis, it will now come into the mitochondria. It will now come into the mitochondria and it will form acetyl coenzyme A. This is something which is called as the link reaction. Okay, it will come into the mitochondria. This pyruvate will now form acetyl coenzyme A. I'm not talking about the anaerobic glycolysis because anaerobic glycolysis is occurring in RBC. RBC तो mitochondria है ही नहीं. तो obviously बात है वहां तो lactate बन जाएगा, ठीक है? But over here, this pyruvate will now enter into the mitochondria, right? And it will now form acetyl coenzyme. So this step, which is linking the aerobic glycolysis to the TCA cycle or the Krebs cycle below, this is called as the link reaction. It is linking glycolysis with TCA cycle. So let us just try to summarize the linking wala part also over here. So in the link reaction, guys, you already know that we have the pyruvate. Okay, link reaction, it is occurring in where? The mitochondrial matrix. The mitochondrial matrix, right? So what is happening? This pyruvate, it is getting converted into acetyl coenzyme A. Pyruvate is a 3-carbon compound. Acetyl coenzyme A, it is a 2-carbon compound. So automatically one CO2, one carbon is lost in the form of CO2. Okay, so this is something which is called as oxidative decarboxylation. Oxidative decarboxylation, right? So you should remember, guys, what is happening. NAD plus is getting converted into NADH plus H plus, right? NAD is getting converted into NADH, right? And this is something called as, okay, the enzyme over here, it is pyruvate dehydrogenase. Pyruvate dehydrogenase. Pyruvate dehydrogenase. Again, this always comes in the form of a 2 marks question in the written exam, guys. So what else you have to over? So this is the reaction that you have shown, but they will ask you the 5 coenzymes of this dehydrogenase. The 5 coenzymes, okay, which are supporting this reaction. The very first one, it is lipoic acid. Okay, the lipoic acid. The second one, it is vitamin B1. It is vitamin B1. It is thymine pyrophosphate. Thymine pyrophosphate. Okay, the third one, we have coenzyme A. Okay, that is the vitamin B5. जब भी हम coenzyme A बोलते हैं बेटा, it is vitamin B5. It is nothing but the pantothenic acid. Okay, it is pantothenic acid. We have NAD plus. NAD plus beta, it is vitamin B3, right? It is vitamin B3. It is niacin, or it is also called as nicotinic acid. Nicotinic acid, right? And we have FAD, right? The FAD plus, that is the riboflavin. It is vitamin B2. It is vitamin B2. So what all vitamins are used over here? Which are the complex of this pyruvate dehydrogenase? We have vitamin B1, B5, B3 and B2. Okay, B1, B2, B3 and B5. Okay, these vitamins are used as a coenzyme for this particular enzyme, that is the pyruvate dehydrogenase. So that is the link reaction, guys. That is the link reaction. So yes, so we have already completed over here. Uh, We have discussed the overview, guys, today. We have discussed aerobic glycolysis. We have discussed anaerobic glycolysis. We have discussed the link reaction, right? I will be continuing with the TCA cycle tomorrow along with gluconeogenesis, right? And I will also try to complete the glycogen metabolism in tomorrow's class. Okay, so I hope you all understood whatever we have discussed today, right? So I'll be coming all to this tomorrow. Okay, so yes, so I hope you have all gone through the core features of this particular app where we are interacting one to one, right? All the notes they are provided in this particular app, right? They're curated in a very in such a manner, no, that you can write the answers just directly by learning from the notes itself, right? And all the plans are given on the screen. So you can just go through it. Even you want a discount, you can use the code Dr. Abhijeet 20, right? For the discounts, right? So thank you for everyone for attending my class with so much patience, right? It's very hard to listen to me continuously for 1.5 hours, but I hope you all enjoyed the lecture. We'll continue the TCA cycle with gluconeogenesis tomorrow, right, guys? So yes, thank you. We'll meet tomorrow, guys, right? So, yes. Ishika, right. Okay, guys. Okay, so shall I end the session, right? Sure. Okay, guys. Goodbye. Thank you so much. Yeah. Thank you. Oracle, the technical team, I think the session has been ended from my side. So just